Okay, next we're going to talk about viruses that infect bacteria. So back in chapter 6, part 1, early on, I mentioned that viruses can infect every type of living organism on Earth. Everything is subject to some kind of viral infection. So there are animal viruses, there are plant viruses, and even there are bacterial viruses, and many more. But being a microbiology class, one of the major topics is to talk about what are called bacteriophages. These are viruses that infect bacteria. So the name is in reference to the viruses that infect bacteria. Sometimes they call them phage for short, but that's in reference to a bacteriophage. I remember learning about these when I took micro, and I just remember looking at a figure just like this and thinking, "There's no way that's real. Like that can't be a real thing." But it is. This is a drawing here, obviously. This is a real picture of a bacteriophage, of the high resolution microscope image. And I've got more. Let me just pull one up on the web here. Here's some other pictures of a bacteriophage. This is, let's see, here's a real picture. That's not the best quality, but that's a high resolution microscope image. They got a little bit distorted. Here's one that very similar to what we were just looking at there. Another one here. This is a high resolution image. It's been artificially colored. That's a pretty good one. Shows the bacteriophage there. I think that one's been manipulated to kind of reorientate those, but those are real images there. A lot of these are computer animations. That's an animation or illustration. This, I believe, is a real one. I could be wrong. I'm pretty sure that's an actual image there. So anyway, these are real things. Here's another real image showing lots of bacteriophages attaching. So hard to believe. I know I was very, very skeptical when I first saw that, but believe it or not, that's a real thing. It looks like right out of a science fiction movie, like a spaceship. What you're looking at is what's called a complex virus. It's got a capsid here, capsid head that has the icosahedral capsid, but it's considered complex because it has all these extra features down here, the tail fibers, the collar, and the sheath. These are all part of the complexity of this shape here. Now, bacteriophages work in a similar way that animal viruses do, in that they have to have lock and key specificity with their host. The virus has to attach to the receptors on the host cell in order to infect. In this case, the tail fibers act as the spikes which attach to the receptors on the bacterial cell. Just like animal viruses, there's a certain range of specificity here. Some have broad specificity, some have intermediate, some have restricted. The difference is they all infect bacteria, so they're always specific to some kind of bacteria, but within the different range of types of bacteria, some are broad, intermediate, and restricted. Now they have a very similar stage, or excuse me, a very similar replication cycle as animal viruses do, and that is they absorb, they penetrate. One difference is that they actually don't have an uncoating stage. So let me come right back to that. So they absorb, they penetrate, that skips uncoating, goes directly into synthesis, followed by assembly. And for some reason I left off one on here. I want you to add to that, they also then release as the final stage. So go ahead and add that there. Assembly is followed by release, just like in the animal viruses. And if you back up, you can look back at the different release mechanisms. Bacteriophages always release through lysis, popping of the cell membrane. So you can add that as well. So release, which is lysis of the cell. So absorption, again, specific to the host, lock and key. They have to match a certain host cells in order to be able to attach, a necessary prerequisite for infection. Penetration in this case is different from an animal virus. Bacteriophages are like a little syringe. You can see here in this figure, when they attach, they literally inject the DNA directly across the cell envelope. So across the outer membrane if it's gram-negative, across the cell wall, and across the cell membrane, and just like a syringe injecting something into your body. That's kind of how a bacteriophage works. And what that does is it just directly injects the DNA or RNA in a naked form, and there's no need for uncoating. So the one difference is it skips the uncoating stage. After that, though, the synthesis, the assembly, and release are very similar as to an animal type virus. Now, one difference here, this is figure 6.19 out of your text. There are essentially two different types of bacteriophages. And they essentially go through two different life cycles and these are in reference to kind of how they might go dormant. So some bacteriophages have the ability to go dormant, like we mentioned earlier with the animal viruses, like those that become a provirus and insert themselves into the host DNA. So back up and look at that if you skipped over that. So there's essentially two cycles that these bacteriophages can go through. Let me skip ahead and come right back to this. These bacteriophages, skip ahead one more time here. Oops, sorry, one more. Where is it? Shoot. Oh, here it is. Excuse me. The word I was looking for was called a temperate phage, a temperate bacteriophage. A temperate bacteriophage, here it says right here. These are bacteriophages that can go dormant in the host cell. Okay, so you have essentially bacteriophages that release them, that replicate continuously. In other words, they enter, they synthesize, they assemble, they release all in kind of one swoop, and that's just considered a normal bacteriophage. Contrary to that, you have what's called a temperate bacteriophage or temperate phage. These are bacteriophages that penetrate and go dormant, and then can release at a later stage. So they don't essentially replicate all in one swoop. They kind of break up that cycle and have periods of dormancy. Okay, so the difference is represented by two different cycles. The normal is called the lytic cycle, and this is showing here where they absorb, they penetrate, they go through the synthesis and assembly, and then release immediately, kind of all in one process, in one kind of short time frame. The alternative to that is where the temperate bacteriophages. What they do is they absorb, penetrate, but then they go into a dormant state. In this case, what's happening is the viral DNA is inserted into the bacterial chromosome, sometimes into the bacterial plasmid. So here it's showing where the pink is showing the bacterial chromosome, and the blue is showing the viral DNA. And what it's showing is that the viral DNA integrates into the host DNA molecule. So it's a lot like we talked about with the provirus. It's really the same thing in a slightly different context. So this is called the lysogenic state, when the DNA integrates into the DNA of the bacterial cell. And now this can basically be a dormancy that can be variable time periods. Sometimes it can be a short period of time. Sometimes it can actually be indefinitely. So it can be anywhere from a matter of hours to it never actually leaves that stage. But a lot of times there'll be maybe a matter of days, or in a lot of cases what happens is the bacteria will replicate the viral information and pass that on to future bacterial cells, and then the virus will pop up later in a future generation. So it's kind of this odd thing that they do to incorporate themselves either semi-permanently or actually permanently into the viral DNA. And that's what's called the lysogenic state. So it's a deviation from the lytic cycle in which they go dormant. They call this the lytic cycle, by the way, in reference to the word lysis, which means to pop and rupture open, which is what you see at the end of the lytic cycle. So they call it that because it ends with lysis of the cell. So lysogenic is in reference to the dormancy that they go into. So that again involves temperate bacteriophages. They become dormant. In this case, the dormant information in the cell is called a prophage. So a prophage is similar to a provirus. This is in reference to the fact that it's a phage virus as opposed to any other kind of virus. And so they call it a prophage. It's basically the bacteriophage DNA inserted into the chromosome, and is then integrated into the bacterial DNA. So the term, a few more terms here. Lysogeny. I know these terms get confusing. Okay, this is where you just got to sit down and commit these to memory. A lot of terminology in biology is confusing, as you already know. So lysogeny is the term that applies to the condition in which the bacteriophage DNA is integrated, the condition in which the bacteria is carrying the bacteriophage DNA. So lysogeny is the condition in which the bacterial chromosome carries the bacteriophage DNA. The term prophage is in reference to the actual DNA of the bacteriophage itself. So they're kind of talking about the same thing under a slightly different context. Lysogeny, bacteria carries prophage DNA. Prophage is the actual DNA itself. Hopefully that makes sense. Induction occurs when the prophage DNA is released from the cell, or excuse me, when the virus is released from the cell. So induction occurs when the prophage comes out of dormancy and continues the cycle. So if we go back here, essentially you can see the bacteriophage can go dormant in a lysogenic state, and then at some point that DNA can be further synthesized and lead back into synthesis, assembly, and then release. So essentially you've got the lytic cycle here taking place like a backwards clock. It can go off into this side state over here in the lysogenic state, and then for various reasons induction occurs and it goes back into the lysogenic state. And that's what we call induction. Lysogeny is a scenario where it actually benefits the virus because it doesn't immediately affect the cell, and it actually gives the virus more time to replicate itself and to pass on that viral genetic information to other bacteria. This is actually part of the context of how viruses evolve over time. The best viruses are those that actually are the least harmful to the host cell. The most virulent, we call virulent, damaging, harmful viruses, those are actually the least successful over time, because you need a host cell to replicate. As a virus, the ideal scenario is that the host cell has no clue that you're there and that you do no damage, and the host cell allows the virus to stay long term. That's really kind of the ideal scenario for the virus itself. When a virus causes lots of damage really fast, it literally kills the very host cell that it needs to survive. Now, of course, they can go on to infect more host cells, but generally that's less advantageous. So lysogeny is this scenario where viruses have kind of evolved to be less impactful and at the very least delay the impact to give them more time to replicate. So when a virus goes into the lysogenic cycle, what can actually happen is the bacteria will make copies of itself and pass that virus on to future generations. So let me just draw this real quick to give you the visual. Here's your bacteria. Here's the phage DNA. Phage DNA being shown here. Oops. Draw this. Being shown here in red. Phage DNA enters and becomes part of the bacterial chromosome. Now, what happens is as this bacteria replicates and makes copies of itself, the copies also have the phage DNA. And as they make copies, they have the phage DNA. And now the bacteriophage is being passed along in more and more cells. Every time this bacteria replicates, another copy of the phage DNA is made, and this bacteriophage becomes more and more common in the population. Now, at some point, induction can occur, and the virus can go back out of the dormancy and pop back up. And at that point, you now have dozens, or even more. In this small example, we've got a dozen or so here that now can all release, and now you've got 10 times more of the viral information that you had prior. If the virus had replicated immediately and caused death of the first cell that it infected, then none of those other cells would have had a chance to spawn, and the virus would have had a much shorter run at replicating. Okay, so I'm kind of generalizing, but that's kind of the idea. If you infect immediately and kill the cell, that kind of squashes your chance to pass on your genetic information to more and more cells. So lysogeny is a condition in which the viruses have kind of evolved to be less harmful in order to increase their success in spread. Last little bit here, one of the interesting side effects of lysogeny, in which bacteriophages go dormant in the DNA, is kind of like we mentioned, some viruses can cause cancer by mutating the DNA. In bacteria, bacteriophages can also cause mutations, and every once in a while these mutations have actually been shown and proven to make some of these bacteria more pathogenic. So essentially, lysogeny can mutate the bacteria, causing changes, and every once in a while that change actually makes the bacteria more harmful. So the infection by a virus changes the characteristic and can make the bacteria more harmful. It can certainly do other things, but this is when we tend to take notice as scientists, as people studying this. we tend to focus on what are the impacts to human health, and this is one of the areas that they're looking at here. So lots of other changes could happen, but one of interest is the fact that they cause a mutation that leads to the production of a toxin or an enzyme that is pathogenic to humans, or it can make something even worse, more pathogenic than it was previously. And the idea, this is called lysogenic conversion. This is when a bacterium acquires a new trait from a temperate bacteriophage during that lysogenic cycle. A few examples that the textbook mentions: Corynebacterium diphtheriae. This is the causative agent of diphtheria induced by a temperate phage. Vibrio cholerae. This is the agent that causes cholera, a waterborne illness that infects many third world countries. And even Clostridium botulinum, the causative agent of botulism. Botulism is a spore forming, or Clostridium botulinum is a spore forming bacteria that leads to a toxin called the botulinum toxin. Also, by the way, what they inject into your face when you get Botox. You can look that up. Botox involves injection of a bacterial toxin into your face or any other part of your body that paralyzes the muscles and the nerves that makes your face go flat. Interesting stuff. Last little bit, prions are not a virus, but mentioned here because they are also a non-living infectious particle. So chapter 6 is mostly about the viruses, but in the end it talks about another type of non-living infectious particle, which is how we defined a virus in chapter 6 part 1, and that is called a prion. Prions are very interesting. They are very rare, but very, very deadly. So luckily they're rare, because when someone picks them up, they are very lethal. And what happens with a prion is basically a protein molecule that distorts other proteins around it. Let me find a figure here just real quick. Okay, so this is showing a type of prion. They give them a name. It's called PRPC. And this is basically a, this is a normal protein, So a normal protein called PRPC. It interacts with a prion, shown here as this little spiky red one, and called the PRPSC. Don't worry about the acronyms. I'm not going to test you on that. But what happens here is that you basically have a normal protein that interacts with a prion, which is basically just a infectious protein. What ends up happening is the interaction between a normal protein and the infectious protein is that the infectious protein converts the normal protein into another infectious protein. So you basically have a protein that converts normal proteins into infectious proteins. It's almost like zombies. How a zombie bites someone and then they become a zombie, and then they go bite someone else and they become a zombie. And yes, I'm talking about zombies. That is kind of like how a prion works. You have a infectious molecule that basically converts other molecules into that infectious form, and then now you have two infectious molecules, and then they go on to infect other normal proteins and convert them to infectious proteins. Now the real issue here is that the infectious protein doesn't function like the normal protein. So there's lots of information here. I'm kind of skipping over lots of detail, but in a sense what happens here is that these proteins affect the nervous tissue. So you have normal proteins in your nerve cells that are doing their normal function, that conduct normal nerve cell activity. Prions are a distorted version of these proteins in the nervous system, and they basically convert normal proteins into non-functional infectious proteins. So when you're exposed to one of these prions, what happens is they slowly convert normal proteins into more infectious prions, which not only stop working and shut down the normal nerve cell function, but then go on to affect more proteins and slowly cause more dysfunction of your nervous system. So you have really two things happening: normal proteins being shut down, they stop functioning, and then a higher rate of conversion of more normal proteins into more non-functional proteins. I mean, the zombie analogy kind of works here because it's essentially one infectious thing infecting more, which then go on to infect more and more and more. So hopefully that makes sense. That's kind of the idea behind a prion. There are two common types. There's really actually quite a few. Prions are not species specific. They can be theoretically passed between any two animals, and that's one of the dangers of them. The disease they cause is called Spongiformencephalopathy. Spongiformencephalopathy basically is a medical term that references holes in the brain. Encephalopathy is a disease of the brain. Spongiform is in reference to sponge-like tissue or holes, kind of like a sponge. So here you're looking at 4 brain tissue samples. Up here in the top left is normal, nice and smooth. Down here is a human condition called CJD, Creutzfeldt-Jakob's disease. And you can see here there are little holes that kind of look like a sponge, I guess. That's how they reference it. And these here are 2 other diseases. Kumaru, which is actually picked up in certain tribes due to cannibalism. Eating brain tissue is actually one way that you can actually pick this up. Super rare, but that's kind of where it originated, actually. And then scrappy is a form that's been seen in sheep and goats and things like that. So they all kind of lead to the same thing, which is holes in the brain. There are different names for different contexts of the disease. Creutzfeldt-Jakob's disease is one of the more common. This is what affects humans and is basically a death sentence. And it's something that when this one gets it, there's no cure for it, and it eventually causes a slow progressive degeneration of the nervous system, and is a very unfortunate disease to be diagnosed with. Luckily, it is very, very rare. Another form of that is what people have maybe heard of before, called bovine spongiform encephalopathy. And if you don't have any clue what that is, it's actually a more technical term for mad cow disease. So mad cow disease was actually a prion disease that you may have heard of. People were talking about this over 10 years ago to a large degree. Mad cow disease was something that started in the United Kingdom due to really, really shoddy practices. They were actually feeding cows that were infected to other cows. They would have a diseased cow that they couldn't sell for human consumption because it was diseased, but there was no law at the time that said that you couldn't grind up that cow and feed it to other cows as a protein source. Yes, they were literally taking diseased cows and feeding them to other cows as a way to save money, and because the cow had value to it and they didn't want to just throw it away, so they ground it up and fed it to the other cows to save money on protein. Yes. And what that did was it basically infected thousands of cows. They went from this being a rare disease, super rare, hardly ever happens, maybe 1 in 1,000, maybe 1 in 10,000. I'm not sure the exact numbers, but it's very rare. I know that. And so very rare. They had probably 1 cow at some point. It was diseased. Should have been just, the body should have been disposed of, and nothing should have been done with it except for probably incineration. And instead, they ground it up and literally fed it to the other cows to save money on protein. Unbelievable. And what that did was it infected thousands more cows. And within a short period of time, the meat supply in the United Kingdom became tainted with prions, and people were eating it and getting sick. And several hundred people died from this terrible disease because of an absolutely ridiculous practice of feeding sick cows to other cows. Unbelievable. But that's how that happened. And that's why the mad cow outbreak took place in the late 90s, early 2000s, somewhere in that time period. Since then, they've cleaned up that practice. They basically had to say, no, you can't do that. You can't feed sick cows to other cows. And they've also eliminated the sale, for the most part, of cow brain, cow eyes, and cow spinal cord. Now, from my understanding, you can still buy those in certain places, but you probably won't. I've never seen them at the supermarket. I think you have to get them from non-conventional sources, let's just say. But generally speaking, it's probably not a good idea to eat cow brain, cow eyes, or cow spinal cord, because that is actually where the prions come from. So if you're just eating the tissue like steak or hamburger, prions are not in that part of the cow. They stay resigned in the nervous system, in the central nervous system, in the eyes, the brain, and the spinal cord. This is also true for any other form of the disease. So if you're eating something that has it, then theoretically you can't get it if you eat just the tissue and not the central nervous system. So one of the problems was they were grinding all this up and creating this meat concoction, kind of hot dogs, which is just a ground up concoction of all sorts of things. So, they had to stop selling those parts of the cow, as well as stop allowing people to feed infected cows to other cows. And that really has led to almost a complete elimination from that in the food supply. So mad cow isn't something that really you should worry about anymore. Creutzfeldt-Jakob's disease, I'll stop it here. I don't want to keep going on this, but look that one up. It's very interesting. That's the human form of the disease. It's actually genetic, and it can actually be transmitted through rare occurrences of like any sort of fluid exchange that involves the central nervous system, which is not very common. So, anyway, that's the last little bit here. As always, email me with any questions. Let me know if anything needs to be re-explained, and I'd be happy to bring it up in class. Have a great day, and I'll see